In this video, we will get started with RStudio and run EasyAmplicon pipeline step by step. Basically, microbiome data analysis is mainly divided into 3 steps and suitable for the analysis of multi-omics data based on second-generation sequencing. Converting big data into human readable graphs and reduce the dimensionality for better realization. Big data is generally raw sequencing data. Each sample is at least tons of MBs, and metagenomic can be up to several GBs or even hundreds of GBs. The data does not need to read. Clustering, comparison, and quantification will be covered in this video. Basically, in this video, we are going to reduce the dimensionality, and we will get from sequencing table to the feature table like OTU and ASV. Fundamental idea of the data analysis includes 3 steps: big data to large data set or big table, which eventually rise to small data set or small table, and then graphs or chart. At present, people rarely read tables, articles only read pictures. We also need to master visualization technology to discover and display outstanding transmission pattern so that peers can understand our finding in a short time and promote scientific communication. The basic idea of amplicon analysis is that we first reduce the dimensionality of 16S data into species composition table, also often called an OTU table, which stands for Operational Taxonomy Unit. Recently, we have started to use ASV tables. 16S data can be pre-functioned and converted into functional composition table of KO and COG. Species composition and functional composition are all character tables. They will also further reduce dimensionality and visualization, making it easier for us to observe pattern. You can see here species composition give rise to through stamp, and functional composition also give rise to pictures. This is the schematic diagram of commonly used structures for amplicon sequencing. You can see this line. The red indicate barcode. Barcode is a sample label which is used to distinguish which sample the sequence comes from after pool sequencing. When more than 100 samples are mixed in the same library, double-end barcode is generally recommended. This is forward primer, and this R stands for reverse primers. Primers designed in the 16S, ITS, 18S conserved region used to amplify part of hypervariable region of ribosomal DNA or ribosomal RNA. Amplicon is amplified ribosomal DNA fragments. Now, via high-seq, nova-seq, or machine parent sequencing, right and reverse complement merge into single end. Then extract barcode sequence, rename sequence by barcode, excision of double-end primers, and amplified sequence amplicon. This is from normally this is the pyramid. You can see here that how we can reduce our big data and which eventually generate feature table, and how the length of the data is getting reduced as it move up. That data is getting reduced. So previously in the last video, we already explained this setting of RStudio we need to adjust before running the pipeline. So we can revise here again. We have to choose UTF-8. We use trend mirror nearest to your location and use bash terminal. Now we will open our RStudio here. Run as administrator. Now the first step, we have to set working directory. And because as we already downloaded Easy Microbiome, EasyAmplicon in our D drive, set working directory, choose directory. When I downloaded the EasyAmplicon and Easy Microbiome from GitHub, by default it downloaded with this name, EasyAmplicon Master and Easy Microbiome. I will open this, and here I can see the pipeline in English, pipeline for Mac user, and this is pipeline in Chinese version. I will open pipeline. As I open this pipeline, now what we need to do here, this is the one which we downloaded from GitHub, but now I am going to run the pipeline, so I have to see what result I am getting from this pipeline. What are the results, what are the output generated by running the code. So here I have to create another folder, either give name amplicon or EasyAmplicon. I am going to give name EasyAmplicon. Now this is empty. Now this is our main working directory here, so I have to set path again here. I will go to the session, set working directory, choose directory, and I'll come here, and this is the one which I just created, this folder, EasyAmplicon. Now see, this is EasyAmplicon. Now this is our pipeline. Whenever we have to start working with this pipeline, we have to set our working directory and software database directory DB and working directory WD. Our working directory is EasyAmplicon and our database directory is Easy Microbiome. So my EasyAmplicon folder, which I just created, is located in D drive, and Easy Microbiome is also located in D drive. So if in case you have in your C, then you have to replace here C. And if you have the name amplicon, just remove and keep the name amplicon. So it's up to you. Whenever you start, you have to run these 4 lines, but in the terminal. So I'm going to open a new terminal here. Now see, when I open new terminal, it is D and EasyAmplicon. So I am going to run these 4 lines to set my working directory and database directory here in the terminal. So starting files. Now I have to create subfolder in this EasyAmplicon. We have to create directory of seek, result, and temporary storage files. Run this code. Now see, you can see here seek, result, and temp. You can open, they all are empty. We need some initial files to start with this pipeline. Metadata, experimental design. We need metadata raw text. So now we will go to the downloaded EasyAmplicon master folder and go to the result, and we will copy metadata raw text from here and keep it in our generated folder result here. Similarly, we will go to the seek folder. If you have your own sequencing, well and good. And if you are testing this pipeline, you just use this sequencing folders. Copy and paste in this seek folder. Now these 2 files we need to initiate our pipeline. We have our sequence file, we have metadata raw text now. Now use CSV to count the number of rows and column in a table. Let's run this code. Number of columns 15, number of rows 18. Now metadata should have at least 3 columns. The first column should be the sample ID and the last column should be the description. Cat command is used to view a file and A option to display symbol, and this symbol is used as a pipe to connect command. Let's run this code. Windows users run the set command to remove and then check with cat A. They will run this code. Now come to the sequencing data. We already created the folder of sequence and have inside sequencing. If user don't have their sequencing data and they want to download it, then they use these links to download the data. But if you already have, then you don't need to do this step. Simply come over here, and it is recommended that user should have their sequencing folder inside the seek. All the sequencing results should have in the seek folder. Now let's view compressed file. We will use this command to compress this. Each line is too long, specified to view 1 to 60 characters per line. Basically, the sequencing results written by the company are usually a pair of FASTQ file or compressed file in GC. So we need to decompress it. So we do this step. And then by using ckit program, we will calculate ckit stat. We can come here. Now see. Now sequencing data, batch count, and summarize table. We will run this command. It gives the statistics of our sequencing The format is FASTA, type DNA, number of sequencing, sum of lengths, minimum length, average length, and maximum length. Now come to the section of pipeline and databases. The database must be decompressed for the first time it's used, and we can skip it later. How to decompress the reference database used by the pipeline? By using gunzip or scikit, we can use search available 16S, 18S, ITS databases including RDB, Silva, and Unite. It's by default saved in Easy Microbiome Win folder. Use search database download page is this. If you have some problem in your use search, you can simply click this link and download from there. And now by using gunzip and zip, we can decompress them. For decompressing gunzip, command will be used. And then ctexstat. To decompress the ITS Unite database, need to download it from the official website or network disk EasyAmplicon use search. Now see, scikit statistic by using use search Unite, format type, number of sequence, sum length, minimum length, average length, and maximum length. Green gene database is used for feature annotation. The default decompression will delete the original file, and C specify the output to the screen, and this command write a new file can be renamed. Now run this code. It gives us the format FASTA, type DNA, number of sequencing is this, sum of length is this, minimum length is this, average length is this, and maximum length is this. And it gives us what to use for. We can check here. We have scikit statistic here. Now come to the next section, read, merge, and rename. Basically, in the next generation sequencing, DNA molecules are fragmented into short pieces, sequenced, and then computationally reassembled. For amplicon sequencing, which focuses on a specific region of the DNA, for example, 16S ribosomal RNA gene for bacteria, the target region is often longer than what can be sequenced in a single run. Therefore, sequencing machine typically used a pair-run approach. The target region is divided into overlapping fragments. Each fragment is sequenced from both ends, resulting in two shorter reads called pair-end reads. These pair-end reads have overlapping sequence and together represent the full amplicon sequence. Merging pair-end reads involve combining the overlapping information from both the read to construct the complete amplicon sequence. This is typically done using specialized software like research mentioned in the script. We can see here. During the merging process, the software can also rename the read to reflect the origin and merge sequence. This helps in organizing and tracking the data throughout the analysis pipeline. For example, the original parent might be named sample ID 1 FAC-GIS and sample ID 2 FAC-GIS. After merging, the combined sequence might be renamed to sample ID merged, which is a clear indication that it presents the merged amplicon. Now we can see here how we do this. There are several methods to perform this. Method 1 is sequential processing with for loop, but some computers do not support rush and scheduling fail during execution. Use for loop section instead. So there is one method for loop and the second is rush. So method 2 is parallel processing with rush, utilizing job count with 2 jobs. The speed is enough. So we will preferably use method 2 to perform this step. It will take some seconds. If compression is not supported, decompress and then merge into parent mode. This is optional, renaming single end files and then integrate sequence after renaming. We will run this code and then now the step is trim primers and perform quality control. In trim primer and perform quality control, amplicon sequencing uses primers to target specific region of the DNA molecule for amplification. These primers are artificial sequencing, not part of the actual target region. Trimming removes the primer sequence from the beginning like from left and right of the reads, sequencing machine might introduce error during the reading process. So quality control aim to identify and potentially remove low quality option of the reads to improve the accuracy of downstream analysis. So we will use this through research, and get bar command we will use it. You can see here, we will run this. It will take some seconds. It emphasized the importance of understanding your experimental design and primer length for accurate trimming, and we search with parameters specifying the amount of bases to trim from each end based on the primer length. And then view the file to understand the FASTA file format. Run this command. The next is remove redundancy and select OTUs and ASVs. Now, the process of removing redundant sequence and keeping only unique ones, sequencing can generate some level of redundancy within a dataset. This can be happen due to sequencing error, high abundance sequencing. So, in the redundancy process of identifying and removing the redundant sequence while keeping the unique one is help us to reduce data size and improving accuracy of our data sets. Basically, to remove duplicates reduce the overall data size, making downstream analysis faster and more efficient. Similarly, focusing on unique sequence provide more accurate representation of the true diversity present in the sample. We can perform this via using research, and high abundant node redundant sequence are very small. We can use this to obtain unique and run this code. All filtered far we see here. This is unique far we obtained here. We come here in the T folder. This is our EasyAmplicon. This is our, we can see here, this is our unique, this is filter, and this is all FQ, and these all are the merged. So come back to the RStudio. Now cluster OTUs or denoise ASVs. Here we are going to discuss two methods for selecting operational taxonomy units, OTUs, or amplicon sequence variant ASVs. 97% clustering for OTUs recommended for large datasets. Denoising for obtaining ASVs recommended for single base accuracy. And for research provide commands for both methods. We will preferably use method 2 to perform this part. Modify sequence name from, you can see it's still running here, and writing Zotus. We have to replace Zotus to ASVs for easier identification. So we will run this code. Then perform reference-based chimera detection. Generally, reference-based chimera detection is not recommended as it can lead to false negatives due to absence of abundance information in the reference database. In contrast, provide two methods for chimera detection using research with RDB database. Fast but prone to false negatives. Not performing chimera detection at all. There are several methods to perform this. First of all, we have to create a folder inside the result with the name raw. And there are two methods to perform this. We will use method 1 using research in combination with RDB. And then use sed command to remove this kind of lines. Now, the next step is feature table. We here get OTUS far. You can see here Zotus far and then OTUS far. Now, the next step is generation of feature table. The principle of generating feature table is amplicon sequencing data typically contain reads representing various DNA fragments within a specific target region. These reads need to be processed and summarized to represent the microbial communities in the sample. A feature table, also known as OTU, which is Operational Taxonomy Unit, or ASV, which is Amplicon Sequence Variant tables, serve as a foundation of downstream analysis. It generates rows and columns with a unique DNA sequence representing organism in the sample. Values in each cell represent the abundance. So how we can generate this? There are several methods to do this. Method 1 is generate feature table using usearch. Method 2 is generate feature table using vsearch. We will use method 2. And the input files, it's required OTUS file, and then it will eventually give us OTU tab text. Let's run this code. And it start calculating here and searching. The core step is generating feature table involved read processing, merging peer and read, trimming primers, and quality control to remove low quality sequences. In de-redundancy, identifying and removing identical or highly similar reads, keeping only the unique one to reduce data size and improve analysis efficiency. Grouping similar sequences into OTUs based on a certain percentage identity threshold like 97%, or resolving them to ASVs, unique sequence without any threshold. Determine the number of reads assigned to each OTUS or ASV in each sample, providing quantitative representation of the microbial community. And after that, we will see here how much it's aligned. This is masking 100%, counting, creating camera index, searching 100%, matching uniquely query sequence is this. We can see here, or now writing OTU table 100%. And we get OTU tab text in our folder which we created. You can see here, inside the result, we created raw and we have OTU tab text. If we click here, you can see now we have ASV IDs under OTU, and then we have our samples here. For Windows user, you should remove the new line character from vsearch result and correct them to the standard Linux format. We will run this code. In order to count rows and columns in a table using CSV take, we will run this command. And we can see here the number of columns 19 and number of rows 1370. Now the next section is species, annotation, and removal of plastid and known bacteria. This step involves assigning taxonomy information, e.g. bacterial species, to each OTU or ASV sequence in the feature table. The script might use a command line tool like usearch to compare the OTU or ASV sequence against a reference database, for example, green gene or silver. Filtering is often applied to remove entries unrelated to the target organism group, for example, plasmid, non-bacteria, or more focused analysis. Species annotation typically output a file containing taxonomic classification for each OTUS or ASVs, for example, taxonomy.txt. We can see here, we will run this code using vsearch, or we used OTUS file, and the output will be OTUS syntax. Let's run this code. Now we can come here, we can see here OTUS syntax. Two methods. Method 1 is the number of original feature table rows. We will click this and then we will run this command, and eventually we will get OTUS syntax discard. And the number of filtered feature table rows, we will run this command and filter the sequence corresponding to the feature table. Filter feature table corresponding to the sequence annotation. And in case if you feel that the screening is not reasonable, you cannot screen, you can use this method as well. And this is the optional statistical method to obtain summary OTUS table. Run this command. We can simply go here, and you can see here we have here OTU tab stat, we have OTU tab stat here. This section is equalization of sampling. The next step is In this step, which aim to normalize the number of sequences across samples to account for potential differences in sequencing depth, the script might use a tool like our script with a custom script out tab rear otu tab rear.r to perform this normalization. The script likely specify a minimum sequencing depth, for example 1000, to ensure a sufficient number of sequencing for meaningful analysis. The normalized feature table might be saved as a separate file, for example otu tab rear text. Now we are going to create another folder inside the result with the name alpha, and by using otu tab text we will generate otu tab rear text, and then we will create here begin text, which will be used in the coming downstream analysis. Run this code all together. Now see here, we will get otu tab rear stack, otu tab rear text, otu tab rear text start, and we generated alpha folder here.